Thank you, Diane, and thank you to Bruce and the organizer. Thank you to Sophian for inviting me to show our research. As Diane said, I am a new faculty member at University of Pittsburgh, so we're still in the process of getting our equipment up and running. So the work I'm going to present you today was not done at UPitt, it was done at NYU with my postdoc mentor, Dick Chen. Today, I'm going to tell you about the modulation of brain activity by cannabidiol, or CBD, and the implications for epilepsy. So you will find that there are several overlapping themes with what Laurie presented, and I hope this can generate some more discussion. As I said, this work was done in the lab of Dick Chen and in collaboration with Dr. Evan Rosenberg, who's now doing his residency at UPenn, and Erica Nebbet, who's now a MSTP student at Stony Brook, and with the guidance also of Oren Devinsky. The motivation of studying CBD initially came from double-blind placebo-controlled clinical study performed at NYU by Oren Devinsky. In those studies, he and his team had shown that CBD can significantly reduce the number of seizures in the intractable form of epilepsy known as Dravet syndrome, and that Laurie beautifully described. Their team had shown that CBD can reduce seizure frequency by up to 40%. And in another pharmacoresistant form of epilepsy, it was known as Lennox-Gastaut syndrome. It was also shown that CBD can reduce seizure frequency by 40%. Another motivation for studying the circuit mechanism of CBD comes from the overlap of the different genes that are involved in brain disorders. We know well that these genes that are involved in several disorders, such as epilepsy, intellectual disability, and autism, and even schizophrenia, strongly overlap, so that perhaps by understanding the mechanism of action of CBD, not only can we know more about the pathology of Dravet syndrome and Lennox-Gastaut syndrome, but maybe it can also be informative for the defects in other pathologies of the brain. CBD is a phytocannabinoid. It is one of several hundreds of different phytocannabinoids that are found in cannabis. It is relatively close in structure to the more well-known phytocannabinoid known as tetrahydrocannabinol, or THC. THC is psychoactive, is produced the classical high associated with the consumption of cannabis, while CBD is thought to be non-psychoactive and does not produce this high. But we know from several studies that CBD has an impact on the brain. And in contrast to THC, CBD does not induce tolerance, psychosis, withdrawal, and hunger, and it is actually thought to counteract those negative effects. The pharmacology of THC is an agonist of CB1 and CB2 receptor, while for CBD, it only has a low affinity at CB1 and CB2 receptors. But speaking of which, the pharmacology of CBD is highly complex. We've seen in seminars and in posters here from Theodore and from Bruce that CBD acts at voltage gated ion channels, such as sodium gated voltage channels and potassium voltage gated potassium channels. And also there is a poster by the Sophian team showing that CBD also acts at T-type calcium voltage gated calcium channels. But CBD is also known to act at G-protein coupled receptors. It is known that it acts at CB1 and CB2 receptor with low affinity and at serotonin receptors. This list is not meant to be an exhaustive list of CBD's target, but I also wanted to highlight that CBD has been shown to act as an antagonist at the G-protein coupled receptors, GPR55. GPR55 was previously an orphan GPCR in the brain, but now we know that his endogenous agonist is called LPI, or lysophosphatidylinosytol. And CBD acts as an antagonist at the GPR55 receptor. Further motivation from this work came from the literature, which shows that there is evidence for GPR55 and LPI signaling in seizure promotion. And that is because LPI, which is a lipid, comes from the more famous molecule phosphatidylinositol. And phosphatidylinositol can be transformed into LPI by the phospholipase A1. In return, LPI can be transformed into phosphatidylinositol by LPI acyltransferase. And it was shown in studies that inactivating mutation that affect LPI that affect LPI acyltransferase are associated with epilepsy, autism spectrum disorder, and intellectual disabilities. So the impact of this would be to increase the concentration of LPI. And our general hypothesis was that LPI could act at GPR55 to have pro-seizure mechanisms, which in return could be counteracted by the antagonistic effect of CBD at this receptor. So today I'd like to show you our results on how does CBD modulate LPI GPR55 signaling to affect seizure. And I will also tell you about what are the neuronal and circuit targets for CBD retuning of hippocampal signaling. The first experiments that we performed was to look at does the anti-seizure action of CBD involve GPR55? And to do these experiments, we used two cohorts of animals that in which we induced seizure with PTZ. We either use wild type and GPR55 knockout animals that were treated with control or with CBD. What we found is that in wild type animals, an increasing concentration of CBD could drastically reduce the number of tonic-clonic seizures that we observed. However, this effect was completely gone in GPR55 knockout animals, telling us that GPR55 signaling was required for the anti-seizure action of CBD. Now we use immunohistochemistry to locate that GPR55 is strongly expressed throughout the brain, but in particular, we found strong expression in the CA1 pyramidal cells of the hippocampus. And there are multiple associations with the hippocampus with epilepsy. So we next looked at how does seizure affect GPR55 expression. By using immunohistochemistry, we observed that the immunohistochemical signal was strongly increased by seizure, an effect which was blocked by the application of CBD. And also, we measure directly the brain LPI concentration, either in the cohort of animals that were induced to have seizure and those that were induced to have seizure and for which we treated with CBD. And what we observed was that in the animals suffering from seizure without CBD, there was an increase in the concentration of the 18O isoform of LPI, which is the form of the isoform of LPI that acts the strongest at GPR55. So that overall, seizure elevates the GPR55 expression and the LPI concentration in the brain. The last experiment that we performed in this first part was to look at how does LPI to GPR55 signaling modify the information transfer in the hippocampus. And for that, we use acute brain slices in which we perform cell-attached recordings from CA1 pyramidal cells and electrically stimulated the classical Schaffer collateral to CA1 pyramidal cell synapse. We adjusted the stimulation intensity to see those action potential waveforms in the cell-attached configuration and then applied LPI. What we observed was that application of LPI in those experiments elevated the action potential probability almost twofold. And this effect was actually prevented by the application of CBD, pre-application of CBD on the slice, and was also completely absent in the GPR55 knockout animals. So that this tells us that LPI increased the synaptically driven spiking probability at the CA3 to CA1 synapse. So in conclusion for this first part, we've seen that LPI act at GPR55, and this kind of promotes excitation in the hippocampal circuit. This hyperactivity in turn, such as a seizure, for example, in our mouse model, can increase the concentration of LPI in the brain and can also increase the expression of the GPR55. CBD can block this interaction to decrease the number of seizures and kind of prevent this pernicious loop of activity in the hippocampus. In the second part of the talk, I'd like to tell you about what are the neuronal and circuit targets for CBD retuning of hippocampal signaling. And I will introduce you to the different types of neurons that are found in the CA1 hippocampus. This is a reductionistic scheme, but the CA3 information is sent to the CA1 pyramidal cell, and there are a host of inhibitory cells that exist in the CA1 circuits. I will focus our attention today on two different types of interneuron known as the PV fast spiking interneuron and the somatostatin expressing regular spiking interneurons. These are two types of inhibitory neurons that release GABA at the synapses that they form, but they are arranged in very different circuit organization. The PV interneurons are recruited in a feed-forward manner by the CA3 pyramidal cell excitatory synapse, while the SST cells are recruited in a feedback manner by the CA1 pyramidal cell. Another distinction between these two types of cells is that the PV cells typically innervate the perisomatic region of the pyramidal cell, while the SST cell innervate the dendritic regions of the CA1 pyramidal cells. These are exemplars of the anatomy of the PV cells and the SST cells, and additional motivation to look into these circuits and into the interneurons themselves came from multiple evidence showing that genes that are involved in epilepsy are expressed in GABAergic interneurons. There's a functional alteration of GABAergic interneurons in multiple forms of epilepsy. It has been shown that GABAergic neurons modulation can abolish seizure activity. And an additional important point is that several anti-seizure medication target the GABAergic system. So despite the fact that the inhibitory neurons represent approximately 10% of the total neuronal population in the brain and in the hippocampus, they have a very strong and powerful effect on controlling the brain network. An experiment that we did was to, again, look at the propagation of activity from the CA3 region to the CA1 region. And in acute brain slices, we delivered short bursts of high frequency activity to the CA3 axon connecting to the CA1 pyramidal cell. And this time recorded in current clamp, the postsynaptic potentials that were associated with this burst of stimulation, first in control condition, and then during the application of CBD. What we observed was that there was a significant decrease in the cumulative action potential probability as a function of stimulus number when we applied CBD. And from there, we thought to look at two things. First, what is the direct effect on CA1 pyramidal cell firing of CBD? And second, how is the propagation of the synaptic activity affected by CBD? So first, we looked at the intrinsic excitability of CA1 pyramidal cells in wild type, in slice obtained from wild type animals. And we measured the number of action potential as a function of the current injection to obtain those FI curves. And we observed that CBD kind of decreased the action potential firing frequency as a function of the current injection. When we perform the same experiment in slice from GPR55 knockout animals, we observe that the CBD could still decrease the FI curves, telling us that CBD does affect CA1 pyramidal cell, but in a manner that is GPR55 independent. So it probably directly affects the different ion channels that control the firing of CA1 pyramidal cells. Next, we looked at the postsynaptic potential that were underlying the firing during those brief bursts of high-frequency stimulation. We observed that the postsynaptic potential were strongly decreased in amplitude by the application of CBD. An effect was completely gone in the GPR55 knockout animals. We wanted to validate this GPR55 dependence, so we used an inverse agonist of GPR55 called CID, and the application of CID mimicked the effect of CBD and further occluded any effect of CBD that was applied after CID, telling us that the decrease in the postsynaptic potential that we see by CBD is GPR55 dependent. The fourth set of experiment, we performed those same recordings, but we applied bicuculin in the control condition so as to fully block inhibition from GABA-A receptor in the slices. And in this case, we saw that CBD had completely lost its effect, telling us that this effect of CBD on the postsynaptic potential requires intact synaptic inhibition in the slices. With that in mind, we use immunohistochemistry, and we found that GPR55 was not only strongly localized to CA1 pyramidal cell, but was strongly expressed in parvalbumin cells, and also to some level in somatostatin expressing cells, so the two types of interneurons that we were looking at. We directly recorded from the two types of interneurons to look at the effect of CBD on PV and SST cells. And in the same stimulation protocol, recordings from PV interneurons in acute slices from transgenic animals, for which we drove the expression of TD tomato in PV cells, revealed that the application of CBD actually increased the recruitment of the PV cells during this stimulation. And again, an effect which was mimicked by the application of the inverse agonist of GPR55. Telling us that CBD increases the recruitment of PV interneuron in a GPR55 dependent manner. And next, we also looked in SST interneurons. But in this case, we observed the exact opposite effect, that in that CBD decreased the recruitment of these cells during short trains of stimulation. Again, this effect seemed to be GPR55 dependent. This is a low end. We're still finishing those experiments. Finally, we tried to track the effect of CBD on these two types of interneurons to different targets. And I'll probably leave you a little bit of a negative note. We didn't find any the targets that were mediating this dual effect. But what we saw was that the effect of CBD was to cause a leftward shift in the FI curves recorded in PV interneurons, telling us that indeed CBD modulated the intrinsic excitability of these cells to increase their recruitment. But in contrast to that, in the somatostatin interneurons, we saw the exact opposite. There was a rightward shift in the FI curve, telling us that these cells became intrinsically less excitable in response to CBD application. Our last experiment was to investigate, can we mimic the effect of CBD by directly manipulating the PV interneurons? And for that, we expressed chanrhodopsin selectively in PV cells and perform whole cell recordings from CA1 pyramidal cell with electrical stimulation of the CA3 afferents, and also time-locked photostimulation of the PV expressing chanrhodopsin in order to boost their recruitment as a function of the excitation coming in from CA3. what we saw was that the photostimulation of PV cell time-locked to the Schaffer collateral stimulation showed the same decrease in the postsynaptic potential that we had seen with CBD. And overall, this photostimulation of PV cell was sufficient to decrease the propagation of firing from CA3 to CA1 in the hippocampus. So that overall tells us that the PV cell increase in activity was sufficient in itself to dampen the propagation of activity, as seen with cannabidiol. So overall, I've shown you today how the anti-seizure molecule receptor LPI GPR55 signaling can be interrupted by CBD to prevent seizure, and I've also and how CBD can prevent this pernicious loop of hyperactivity by antagonizing GPR55. And I've also shown you that in the hippocampal network, CBD dampens the propagation of high frequency activity. And it does so by acting directly on the CA1 pyramidal cell and modulating their intrinsic excitability downwards, but also it has this counter-modulation effect on PV cell and SST cells, which we have shown that the modulation of PV cell was sufficient to dampen the information transfer from CA3 to CA1. 15 seconds. Shameless promotion for my lab. I just started my lab at the University of Pittsburgh. I'm looking to hire at all levels, so please get in touch with me if you're interested in these studies. And our main focus is to understand how different population of interneurons cooperate in the hippocampus in order to allow learning and memory. And finally, I'm happy to take questions. I'll leave the conclusions for study here to facilitate discussion. Thank you.
Thank you so much. I think we can probably take one really quick question, and then you can all gather around Simon and ask loads of questions as we're eating. But is there a quick, go ahead, Bruce. Yeah.
Really interesting, Simon. Do you know with the GPR55 what it works on, what it's coupled to?
It signals via G alpha, and after that we looked at potential signaling cascade. We didn't find anything that would, and I think in our case, what we were looking for is a signaling cascade that would be specific to PV cell versus SST cells, and we didn't find anything specific. So from then, we stopped looking at this hypothesis. It's very hard to reconcile while the same molecule is this two different effects on cells that are very close to each other in the brain, acting via maybe the same receptor. Or we're also thinking that not only is GPR55 a target, but there are multiple other targets of CBD, including direct modulation of ion channels, of course.
Great. Well, thank you again. Simon, you're going to be available at lunch. Yes.
Absolutely. Thank you.
Lots of questions, but thanks to all the speakers this morning. Thanks.